Hi, good morning, everyone. Thank you for joining this plenary talk. My name is Yu Hao. I'm the professor in National University of Singapore in Biological Sciences. Today, I will serve as the moderator for this talk. Uh, First of all, let me introduce our speaker, Professor Thomas Cech. Professor Cech was raised and educated in Iowa, earning his Bachelor of Arts in Chemistry from Grinnell College in 1970. He obtained his PhD in chemistry from the University of California, Berkeley, and then engaged in postdoc research in the Department of Biology at MIT. In 1978, he joined the faculty of the University of Colorado Boulder, where he became a Harvard Hughes Medical Institute investigator in 1988 and the distinguished professor of chemistry and biochemistry in 1990. In 2000, Professor Cech moved to Maryland as president of the Harvard Hughes Medical Institute, which is the largest private biomedical research organization in U.S. In 2009, Professor Cech returned to full-time research and teaching at the University of Colorado Boulder. Professor Cech's work has been recognized by many national and international awards and prizes, including the Henneken Prize of the Royal Netherlands Academy of Science, the Albert Lasker Basic Medical Research Award, and the Nobel Prize in Chemistry 1989, and also the National Medal of Science in 1995. In 1987, Professor Cech was elected to the U.S. National Academy of Science and also awarded a lifetime professorship by the American Cancer Society. In today's talk, Professor Cech is going to discuss CRISPR revolutionized research. We know now with CRISPR, we can knock out genes quickly and precisely, and also more interestingly, you can even add tags that allow us to follow individuals' molecules in living cells to determine their dynamics and function. So without further ado, let's welcome Professor Thomas Cech to discuss this topic. Professor Cech, please.
Thank you, Yuhao, for that very nice introduction. And let me start by saying that I have had a very nice week at the GYSS. I thank all of the students who came to the session with me, the small session, where we had, I think, some good conversations. And I was quite amazed by the variety of countries that were involved. It was almost the case that no two students came from the same country. Now, I'm going to do something a bit brave here today because I pre-recorded my talk, and instead of using the pre-recorded version, I'm going to go live here. So we'll see how this works. And could someone tell me if you can see my slides okay?
It's okay.
Thank you so much. I needed that reinforcement. Okay, so today I'm going to talk about the topic of CRISPR genome editing and the way that it has transformed biomedical research. And this is a very timely topic because, as you know, the Nobel Prize in Chemistry was just awarded to two women, Emmanuelle Charpentier from Germany and Jennifer Doudna from the University of California, Berkeley, for their development of CRISPR genome editing. So we had a chance to talk, Jennifer and I, a couple of days ago on a panel discussion about the medical possibilities. Today, I'm going to talk more about the research possibilities. So first of all, what does CRISPR do? It cleaves DNA, but of course, it's not as simple as this very primitive diagram. Instead, the CRISPR carries along with it an RNA molecule, and that RNA searches the genome, the DNA, for a region where it can base pair with the DNA. And when it localizes that particular region, the Cas9 enzyme that is also part of the complex cuts the DNA on both strands, and so the result is a double-stranded DNA break. Now, the rest of the editing is done by the cellular machinery. It is the human cell, for example, that repairs this double-stranded DNA break by one of two mechanisms. It can either repair it by a process that the technical term is non-homologous end joining, which leads to some little mistakes in the DNA near the repair. Those are called indels. Or if there is a donor DNA provided, then that can be used to repair the cut DNA, and you can put new sequences into the genome at this point. Now, the medical applications we talked about a couple of nights ago, so I'm not going to go over this again. Certainly, there are some technical challenges. There are some diseases such as genetic diseases that might be an early place where this could be applied, and the ethical concerns must always be discussed and talked about in great detail. And some experiments such as germline editing are, we think, premature to do at this point. But let's talk about the research applications. So because we can now surgically precisely edit the human genome, we can do three things, which I will give examples of from my own lab's research. We can inactivate a gene to help understand its function. We can edit a single base pair in a gene to repair a mutation, or we could create a mutation. And we can add sequences encoding a fluorescent tag, and this allows us to follow a protein as it moves around and explores the environment of a living human cell. We can look at the dynamics by which proteins find their home inside of our cells. Now, the examples that I'm going to be using from our own research have to do with RNA, because we are a ribonucleic acid lab, and particularly not the ribozymes that we originally discovered were the catalytic RNAs, but something that was probably evolutionarily later, ribonucleoprotein enzymes. And we know that these are biologically important because they are still around today. They are not just ancient complexes, but the ribosome, for example, is a ribonucleoprotein enzyme. And this RNase MRP that I'm going to talk about here, that my student Kate Goldfarb worked on, is another example. So she wanted to know whether this gene was really functioning in ribosomal RNA processing. And to do this, she wanted to inactivate the gene in a living cell. Well, CRISPR is really good at this. So here is a diagram of the RMRP gene shown in red. And you can see that if you would use guide RNAs to cut, for example, at positions 1 and 4, then that would delete most of the gene. And then when position 1 was healed to position 4 by the cellular machinery, by the non-homologous end joining, you would have a shorter part of the chromosome then, and it would no longer be able to make this MRP product. Also, you could cut at positions 2 and 3. And again, you would remove a large portion of the gene and thereby inactivate it. You can use polymerase chain reaction to show that if you use these guide sequences, you in fact do get a smaller product, smaller by the amount that you would expect if you had deleted the middle part of the gene. So that's very successful. This is in the whole population of cells, so it's very efficient in this particular case. Most of the cells have been edited, only a small number have not. These other clones caused by cutting at just positions 1, 2, 3, or 4 look like there's been no change in the size. But if you sequence across the gene, you get a clean sequence in the unedited case, but if after the genome editing, you get a clean sequence and then you see that there have been some insertions and deletions which cause the sequence to be scrambled after that. So even though these look intact, they are in fact have a problem because of this insertion of extra sequences during repair. So this was just very brief to show you how you can inactivate a gene. Research goal number 2 would be to edit a single base pair in a gene to repair a mutation. You can think about this as using a word processing program, and you're typing perhaps your PhD thesis or your master's thesis, and you make a mistake, and you go back and you can change that very easily on the computer. Well, now we can change the alphabet of the DNA, the A's, G's, C's, and T's, very easily, and even change just a single one of those letters to a different letter. So we use this, we developed a genome editing strategy with CRISPR and the Cas9 nuclease to understand something about telomerase action in human cells. So how did this work? Well, first of all, what is telomerase? This was discovered by Liz Blackburn and Carol Greider, who won the Nobel Prize, shared it with Jack Szostak for this discovery. And it's a very interesting enzyme because it's not just a protein, but it's also an RNA molecule. So it's a ribonucleoprotein enzyme. Among other things, the RNA provides the template for the DNA sequences being added to the end of the chromosome. In this case, it's the sequence from Tetrahymena thermophila, the ciliated protozoan, the pond animal from which telomerase was originally isolated by Greider and Blackburn, and it has a sequence, 2 T's and 4 G's, repeated over and over again. So this enzyme can add this sequence, ratchet back, add it again, and add multiple copies of this 6 nucleotide sequence. The RNA cannot do this by itself. It needs a protein partner, which you might be able to tell from the name is a reverse transcriptase, sort of like the HIV reverse transcriptase. Instead of going from DNA to RNA, it uses RNA to template DNA. And this was discovered in Boulder, Colorado by Joachim Lingner, a Swiss postdoctoral fellow. And it was only when we could combine the protein with the RNA that we could reconstitute, or that other labs also could reconstitute, the telomerase activity in the test tube. Now, this is medically very interesting because it's kept in a very tight balance in our cells. If you have too much telomerase, this can be a step towards cancer because 90% of human cancers involve the reactivation of telomerase. If you have too little of it, then stem cells have trouble continuing to divide, and you can get a number of diseases of stem cell insufficiency. So let's go back to the cancer question. This telomerase reverse transcriptase, which has to be activated in human cancers, seemed like a very strange cancer-causing gene or oncogene, because usually oncogenes are marked by gene rearrangements or mutations that in the open reading frame that change their amino acid sequence. And the TERT was very rarely associated with gene rearrangements. So what was the mystery? How was this solved? And it took about 10 years after Blackburn and Greider's original discovery, in fact, more than 10 years, for 2 laboratories, Levi Garraway's lab at the Dana-Farber Institute in Boston, and a group at the German Cancer Institute in Heidelberg, Germany, to find out that everyone had been looking in the wrong place for the mutations. They were looking in the coding sequence, and instead the cancer-causing mutations were a little bit upstream from the start site for the TERT gene. Not very far, but just enough that they eluded discovery for many years. And what did these mutations do? They're shown in red here. They created new binding sites for a transcription factor that recognizes this TTCC sequence, and that allowed additional upregulation or transcription of this TERT gene. Remarkably, this tumor-specific point mutation, which was originally found in melanoma, is later found in brain cancers and bladder cancer and liver cancers and many other kinds of cancer. And it is now known to be the third most frequent mutation in all of cancer, following only KRAS and P53. Very interestingly, a number of other very common cancers, including prostate and breast cancer, do not have this mutation, but they still upregulate TERT. They still find a way to make more of this TERT and make telomerase, but not through this particular mutation. And that's still a bit of a mystery. Maybe some of the students in the audience can solve that for us in the future. So we were able to develop, now, unlike the first case that I talked to you about, this genome editing was extremely inefficient. This part of the human genome is not very accessible to the CRISPR machinery. And so we had to look through hundreds of cloned cells in order to find one that had a mutation. And the students got very tired of having to do all of this work in the tissue culture lab. So they scratched their heads a bit and did something more clever. The idea was, and this was developed by Ling-Hei Shi, graduate student in the laboratory, was to incorporate in the donor template a green fluorescent protein gene in addition to the mutation that we wanted to introduce. And then we could, after genome editing, find the very rare cells that were fluorescing green and keep those and throw the rest away. But of course, we didn't want the green fluorescent protein in the genome at the end. So we did another round of CRISPR genome editing to remove the GFP, but to retain the new mutation that we had added. And now we do cell sorting for cells that are not glowing green, that are not fluorescent. And this allowed us to purify cells that had this single change in the human genome, base X or base pair X changed to base pair Y at a particular position. So there's too much technical detail in this slide. I apologize, but this is just some data that validate the fact that when we repaired this TERT promoter mutation, which you will remember was a activating mutation, that we then had, when we repaired it back to the normal base at that position, we got less telomerase activity, as we would expect, about 50% less, because only one of the two copies of the gene was corrected. We got shorter telomeres, and we got slower growth of the cells. So we could see the function of this mutation directly. Now, the last application that I'm going to talk about for the CRISPR genome editing is to insert a little light bulb that will glow, a fluorescent tag into a protein, and this will enable us to follow amazingly single molecules of a protein around the cell nucleus in live cells in real time, okay? And this was work done by Jan Schmidt and Art Zog in the lab. The particular thing I'll show you started out in 2016, 2017, and this is the live cell imaging of telomerase. So the question we were asking and it's always important in science to ask questions rather than just to do experiments, was how does telomerase find the telomere within the complex environment of a human cell? Now, what is a telomere? Well, a telomere is the ends of our chromosomes, and they consist of double-stranded DNA. I already told you about the 6 nucleotide repeats. They are double-stranded through most of the telomere, and then at the very end, there are a few hundred nucleotides of single-stranded DNA, which we call the tail at the end of the chromosome. And both the double-stranded and single-stranded parts are bound by a protein complex that Tizia DeLange at the Rockefeller University in New York City has called the shelterin complex. And in addition to sheltering and protecting the chromosome end, the shelterin complex also recruits telomerase by a protein-protein interaction. So what Jens Schmidt in my lab did was to put sequences encoding this fluorescent tag called the flag tag attached to the TERT gene right in its normal spot in the chromosome, and also to attach a different color fluorescent tag to a protein that binds to the chromosome end. And then, just so because he likes lots of different colors, he put blue fluorescent protein on a protein that lights up the Cajal body, which is a nuclear organelle that is involved in maturation of the telomerase complex. Now, the movie I'm going to show here is 12 seconds in the life of a HeLa cell nucleus. Here is the nucleus of the cell. The red dots are the chromosome ends, and you will see that they don't move very much in 12 seconds. They're pretty static in the nucleus. The green dots are the fluorescent single molecules of telomerase, and the blue is the Cajal bodies. And you will see that the telomerase molecules are moving, diffusing around the nucleus with amazing kinetics. Look at those dynamics. Now, I'm going to play the movie again, and this time I want you to, instead of being mesmerized by all of the dancing telomerase molecules, I want you to focus on this little spot that looks yellow, because this is a place where telomerase in green has found a telomere in red, and they stay together for the entire time of this movie. And those, we think, those rare interactions are long enough for telomere extension. Now, you can't just look at the pretty pictures and publish a paper. You need to track many thousands of these molecules and look at their diffusion shape and determine diffusion coefficients. And we saw that each of these different colored lines is a single 12-second pathway of a single molecule in the human cell nucleus. And you can see that most of the nucleus is explored, but not evenly. In fact, the nucleolus, which has no telomeres, is not explored by telomerase. And so I won't have time to show you data that we collected, but the model that came out of this was that telomerase probes the end of the chromosome thousands of times during the S phase of the cell cycle. And then only infrequently does it lock down on the very end. And this stable interaction requires both protein-protein interaction between the shelterin complex and the TERT protein, and also base pairing between the end of the chromosome, shown as this dotted line. Remember, that's single-stranded, so it's accessible for base pairing, and the telomerase template. Now, to test this model, and it's always critical to test our models, we were able to obtain an antisense oligonucleotide being developed as an anti-cancer drug by Geron Corporation. And this antisense nucleotide pairs with the template region of the telomerase RNA. And if our model is correct, then having this tied up with the red strand is going to prevent it from docking onto the end of the chromosome. And indeed, when we had no drug, we had a certain amount of these long static interactions. About half of the cells had at least one long static interaction. But when we added this antisense oligonucleotide, we got a statistically significant, rather large drop. And at intermediate concentrations of the drug, we saw intermediate amounts of this long static interaction. So that supported the original model. So the conclusion then is that human telomerase finds the telomeres in two different interactions. One, a base pairing between the RNA in the telomerase and the single-stranded DNA at the end of the chromosome, and the other one being this protein-protein interaction. Telomerase conducts a rapid three-dimensional search of the nucleus, probing each telomere thousands of times in the DNA synthetic phase of the cell cycle, but it locks down on the very end of the chromosome, the 3' terminus, only rarely. Now, if I had more time, I would talk about our more recent work with epigenetic silencing of genes, and I would have time to show you more movies of different molecules now screaming around the cell nucleus, but also engaging in binding to the chromatin quite often, and also show you how we could make mutations in this complex, and that the mutations very much affected the amount of time that these chromatin modifying complexes are sitting on the chromatin and doing their job in the nucleus. But alas, I promised the organizers that I would leave plenty of time for your questions, so I am not going to have time to do that part, but I instead will just summarize by saying that I have described 3 different kinds of research applications of CRISPR that we and others have done in the laboratory. And they all arise from the ability of CRISPR to surgically precisely cut the human genome. The 3 applications that I discussed are inactivating a gene, cutting out a big piece of it to help understand its function, editing a single base pair in a gene, something that would have been thought of as extremely science fiction 10 years ago, but now is done routinely, editing a single base pair in a gene to repair a mutation or to introduce a mutation, and then adding sequences that encode a fluorescent tag, which I showed you in the case of the telomerase, which allow us to follow a protein as it explores a living human cell and test different models about how it is recruited to its site of action. Finally, I would be remiss if I did not give great thanks to the people in my laboratory who have come from many different countries and a very diverse group that have worked on telomerase. Jan Schmidt's work I showed, he's now on the faculty of Michigan State University. Ling He is now at Rockefeller. The epigenetics project that I didn't talk about, done mostly by a graduate student, Dan Yeomans, and the RNase MRP work done by a different graduate student, Kate Goldfarb. And we thank our funding agencies, and I would like to thank you for your attention.
Thank you very much, Professor Chet, for this nice talk. Now we actually have a few questions from our audience. The first one is pretty practical questions. is regarding a GFP gene. So when you add the GFP gene to the DNA sequence, how do we ensure that this GFP will be expressed in the cells, and how do we ensure that the cells actually contain this insertion will show the fluorescence?
Yes, so we had to test and make sure that the GFP was expressed in the cells, and that when we transfected it in, that it would glow and the cells would be green. So that's an important early study before we did the critical experiments. And then how did we know that it was in the right place in the genome? Well, you cannot assume that either. And so we had to PCR amplify the TERT gene and show that that fragment was larger, and then actually sequence it and show that the GFP had been inserted properly. And then we had to do that again after we removed it by CRISPR genome editing. We again couldn't assume that the cutting out was precisely correct. We had to amplify and sequence individual clones to make sure there were no mistakes introduced through this complex process. Thank you for that question.
Yeah, so I hope Professor Cech's answer actually replies to Lulian's question. The second question is regarding, I think, is the underlying mechanism of the hereditary disease. So the question is, how do scientists actually know which DNA mutation actually corresponds to a specific hereditary disease? So if you don't have knowledge in these sort of things, when you do the genome editing, that could be you don't really have very specific targets that you can work on, right? So
Yes,
I guess that Professor Cech certainly can give you some ideas about that.
yes, that's a very good point. And of course, for medical applications, we would start with the CRISPR genome editing in those cases where we do understand the molecular basis of the genetic disease. I'll give you two quick examples. Sickle cell anemia, where there is an amino acid change in hemoglobin gene. That has been studied over and over and over again, and we know for sure that that disease is caused by that mutation. Or muscular dystrophy, in which case failure to make dystrophin leads to terrible early death of children who have this genetic disease because of their failure to be able to have proper muscle function. And so those would be the places we would start. Schizophrenia, autism, many other diseases that have a genetic basis. It's very complicated, and scientists, it's probably 100 or several hundred different genes which, when mutated, can predispose to those diseases. We certainly would not start trying to cure those with CRISPR genome editing in the near future.
Yeah, thank you, Thomas. So the next question, I guess, is really a lot of people who are working on CRISPR systems will be very interested in this question. I guess it's regarding how can you decrease off-target genome editing?
Yes, well, there are a number of tricks that have been used for that and have been published, and it is a very technical question. So I would just urge you to read the literature. I will just summarize by saying that by using different CRISPR systems from different bacteria, and by adding various drugs, and one can reduce the amount of off-target editing, but we should never assume that we have a pure system and that we have eliminated it completely, because there probably is always some low level of off-target editing. People are still working to minimize this.
Yeah. Okay, thank you, Professor Chet. The next question is regarding the telomere size. So the audience asks whether there's a well-established knowledge on the ideal or normal telomere size in humans. If we know that, then probably we can use CRISPR as a tool to keep the ideal size, so that we can live longer, right? Yeah. So, Professor Cech, please. Yeah.
Yes. Well, I could talk for 1 hour on that topic, but I will not. It appears that telomere size does not need to be precisely regulated. It is controlled within a range of sizes. Let's say between 4,000 and 8,000 base pairs, or so, and then that's fine. If you're in that range, it's fine. Now, as you get older, telomeres do shorten because telomerase is no longer active. And if you get critically short telomeres so that they can no longer protect the end of the chromosome, then you get genome instability. So, and that can cause cancer or it can cause cell death. So really short telomeres are sort of a danger. And then the question of whether we can use CRISPR to adjust the size of telomeres. Remember that the telomere is a repeated sequence. And so this might be one of the most challenging places to employ CRISPR genome editing, because the machinery wouldn't know where along the telomere to sit down because it all looks the same. And so it's much easier to design experiments to genome edit a unique sequence than it is a highly repetitive sequence.
Thank you, Professor Cech. The next question is, do you think that CRISPR can be used to solve our problem on antimicrobial resonance? It's a very broad question.
Yes.
Yeah.
The resistance to antimicrobials is mostly due to changes in the bacterial genome that make it resistant to the antibiotic, right? So CRISPR genome editing of the human would be unlikely to solve this problem unless you could, for example, enhance the human immune system and make it better able to eradicate infections. And in terms of applying CRISPR to bacteria, I would say good luck with that one, because there are zillions of bacteria inhabiting the earth. Mm-hmm. And the idea that one could target CRISPR to all of them to reduce their ability to be drug resistant, I would boggle my mind, okay? But I would encourage the students, some of your students out there and the person who asked the question may be smarter than I am. And so keep thinking about this and maybe you can come up with a way.
Yeah. So I guess a CRISPR system, in fact, by nature, is the innate system in bacteria, right? So it's part of the system in bacteria. So there will be different versions of things that human probably can work on, different versions. Yeah. Well, yeah. The next question is is regarding for the clinical approach. How do you target cancer cells using CRISPR in human body?
Yes, I think we talked about this with Jennifer Doudna 3 nights ago in one of the first sessions, and she and I both agreed, and again, we may be wrong, but we both agreed that this is going to be really tough because to use this for an anti-cancer approach, because CRISPR editing is unlikely to be 100% efficient, right? Boy, in our lab, if it's 90% efficient, that's a really good day. So let's say by some miracle, an oncologist could get it to be 90% efficient in editing tumor and killing tumor cells. Well, the problem is that the other 10% of the cells that weren't edited would, within days or weeks, make the tumor again. And so it's a tough project to try to eradicate 100% of something. I think that the applications for CRISPR are going to be more readily understood in areas where we can correct, where we want to have a gain of function, and we can correct maybe only a third or a quarter of the genes in muscular dystrophy or in sickle cell anemia, and maybe that will be enough to switch over to a healthy person.
Professor Cech, there's another question. I think before we close the session, I guess many audience and even scientists probably concur with this concern. Is regarding when we use this new technology, that could be the ethical and the regulatory issues, right? So
Yes.
I think one audience has a raised issue when we use CRISPR, whether we can actually take into consideration the relevant ethical and the regulatory issues. Yeah.
If we're going to apply to humans, we of course must. And if we're going to apply to animals or plants, we must as well. But what I was talking about today was using it in the lab for research purposes. And when we're looking at single cells on a petri dish, we do not consider that we have an ethical problem with doing the genome editing. More broadly for society, I think it's critical to think about this. And Jennifer Doudna has been a leader in thoughtful consideration of the limits that should be put on CRISPR genome editing. And she and I and many others think that it is premature at this time to consider germline editing, which would be inherited by the next generation. Somatic cell editing, I think fewer ethical problems. Juhao, if I may, I would like to just close with a comment.
Yeah, sure, please. Yeah, Professor Cech. Yeah.
Thank you. So when I talked to the young scientists at the GYSS a day ago, many were interested in thinking about what kind of career they should take. And so clearly there are a universe of ways to use your scientific talents and your passion for science. And for me, being an academic and a faculty member has been a very satisfying career. But others have found, satisfaction in the pharmaceutical industry, the biotechnology industry, or teaching, or writing about science, or editing in scientific journals. And so what I would say to the young scientists here is simply, you need to find your own way to use your scientific talents and passion. Thank you.
Thank you very much, Professor Cech. I would like to apologize for our audience that because of the time constraint, we are not able to address all of the questions raised by our audience. I do see a lot of questions, other questions, which are excellent questions. And maybe Professor Cech and myself, we can discuss later after the session is over. But please, I think, please join me to thank Professor Cech for this very nice talk and for his encouragement for all our young scientists, or even now maybe you are not young scientists, but potential young scientists on your way to be, either researchers or to actually achieve your dreams to be a scientist, right? So thank you very much, Professor Cech, and thank you very much for our audience. Yeah, thank you.